आज हम बात करेंगे आरएनए प्यूरिफिकेशन एक्सट्रैक्शन की दूसरे सैंपल टाइप से। जैसा कि आप पहले से जानते हैं, किसी भी टिश्यू से आरएनए आइसोलेट करने के लिए हमें किन प्रोसेसेस से गुजर के सैंपल प्रिपेयर करना पड़ता है। उसी तरह से आज हम सेल्स के हवाले से बात करेंगे विच आर ग्रोन इन एनी सस्पेंडेड फॉर्म। सेल कल्चर, जिसको हम इन विट्रो सेल कल्चर भी कहते हैं, इन दिस केस वी हैव टू ऑप्शंस टू कल्चर द सेल्स, वन इन द फॉर्म ऑफ सस्पेंशन, द अदर वन इज इन द फॉर्म ऑफ मोनोलेयर कल्चर। अभी सैंपल प्रिपरेशन के लिए वी आर गोइंग टू टॉक अबाउट द सेल्स ग्रोन इन सस्पेंशन। जैसे कि मेथड के नाम से जाहिर हो रहा है कि ये सेल्स जो हैं ये सस्पेंडेड फॉर्म में कल्चर किए गए हैं, और अगर हमारे पास कोई वॉल्यूम लिक्विड का है, दे आर सस्पेंडेड इन दैट, एंड टू सेपरेट दीज सेल्स फ्रॉम द कल्चर मीडियम द बेस्ट possible method is to apply the centrifugation method. In this case, we are going to centrifuge these cells suspended in the culture medium. The speed of centrifugation varies from 200 to 1900 G. It depends on your suspension density or the cell types. You decide which speed and what time you need for this centrifugation step. The time will be for 5 to 10 minutes and the temperature will be the room temperature. So the cells suspended in the culture will be subjected to centrifugation for 5 to 10 minutes at room temperature. This time is sufficient enough for all the cells to settle at the bottom and to make a pellet. Whatever is left behind in the tube is the culture medium, which is also known as supernatant, and it is no more required in this process. That culture medium which is not required, the cells is settled at the bottom in the form of a pellet. We are going to aspirate the culture medium. Aspiration means you can do it by using a simple pipette and pipette filler, but to get a best removal so that no traces should be left behind and the process should be speedy enough, we use vacuum aspirators. इस पिक्चर में अगर आप देखें, ये एक एस्पायरेटर की पिक्चर है. दिस पर्टिकुलर कंटेनर इज द लिक्विड स्टोरेज कंटेनर. Then this needle shaped structure is basically the aspirator needle, which is normally touched to the liquid from the tube which you want to aspirate, and this system is connected with a vacuum pump. एस्पायरेटर सिस्टम कैसे काम करता है उसको समझने के लिए अगर हम इस पिक्चर को देखें तो हमें इसके कंपोनेंट्स के साथ साथ पता चलता है कि हाउ इट इज कनेक्टेड विद ईच अदर. द टिश्यू फ्लास्क कल्चर इज द कल्चर फ्लास्क in which we have the cells in suspended form, or we have the cells which we want to separate from the culture medium, और इससे हमने एस्पायरेशन करनी है. This culture flask is linked with a aspirator needle which will suck all the liquid from this culture flask. The second next flask is called as collection flask. The collection flask is going to collect all the waste which is aspirated from any of the tube or any of the reaction mixture. Then we have another flask which is called as liquid trap. Liquid trap is a kind of safety flask in case if the volume of liquid in this first collection flask is maximum and the liquid moves towards the vacuum pump, this liquid trap flask will trap the liquid and will not let this waste to go into the vacuum pump and to block the vacuum pump. Then we have in between a HEPA filter just to act as a filter so that the vacuum system cannot be disturbed, and all these three things are linked to the vacuum house or vacuum pump. Basically we apply vacuum on the system, whole of this aspirator system, and under the influence of vacuum the aspirator needle is going to aspirate all the liquid or all the culture medium from the culture flask. Once the liquid medium is aspirated, the next thing is washing of the cells. For this purpose we add PBS. This is a buffer, the phosphate saline buffer, which is used to do the washing. And after simple washing or mixing of the cells in this PBS buffer, we are going to repeat the step which we did for the culture medium. Means we are going to do the centrifugation. The centrifugation process is going to let the cells settle at the bottom in the form of pellet. The PBS as supernatant is no more required. We are again going to aspirate this PBS from the tube. And the next is the addition of solution D, denaturing solution, which is added at a volume of 2 milliliter for every 1 million cells. So you can count how many cells you have here using hemocytometer method, जो कि हम पहले बात कर चुके हैं. And according to the cell density, we choose how much volume of solution D is added in the tube for cell homogenization. The next step would be the last step to prepare the cell lysate. This is to homogenize the cells. As you know, animal cells की जो outermost boundary है, that is cell membrane. To get everything out of the cell membrane, we have to rupture the cell membrane, and that process is called as homogenization. Homogenization के लिए हमारे पास polytron homogenizers हैं. We can use these polytron homogenizers for 15 to 30 seconds at room temperature to lyse the cells, and at this step, all the cells are lysed. All the RNA molecules are out of the cells in the cell suspension or cell lysate.